In this video, we're going to look at how the embryo develops using Xenopus as the model system. Frog oocytes are asymmetric, and by that I mean that the top half is pigmented while the bottom half is white. We refer to the pigmented pole as the animal pole and the non-pigmented pole as the vegetal. And this pole, the vegetal, when compared to the animal pole, contains less active cytoplasm and most of the yolk. So the oocyte has one line of asymmetry between the two poles. However, when you look at the mature animal, it has more than one line of asymmetry. It has three: left and right, anterior and posterior, that is top and bottom, and dorsal and ventral, that is back and front. So how does the oocyte manage to go from 1 to 3 lines of asymmetry? The Xenopus oocyte is a great, and the pun is intended, cell to use to explore development, as it is big. In fact, in cellular terms, it's very big. The total volume of the Xenopus oocyte is about 1 microliter, or 1 billion cubic micrometers. In comparison, a human oocyte has a volume of around 4 million cubic micrometers. In this poorly drawn diagram of a Xenopus oocyte, you can see the two poles, the pigmented regions of the cytoplasm, and also the localization of the messenger RNAs VegT and Wnt11. For the sperm to fertilize the oocyte, it needs to bind to a receptor, and there are more receptors in the animal pole than in the vegetal. And the point opposite where the sperm binds and enters the oocyte will become the dorsal, the upper side of the frog. Once the sperm has entered the oocyte, it migrates towards the oocyte pronucleus. The egg cortex, the pigmented layer just below the surface, will rotate 30 degrees and give rise to the grey crescent. And the region around the grey crescent is destined to become the dorsal surface. Hence, at the point of fertilisation, we start to establish the other lines of asymmetry. The single Xenopus cell then undergoes a series of 12 rapid synchronised cell divisions, which over 7 hours produces a blastula, a hollow ball of around 4,000 cells. At this stage, the ball of cells is still the same size as the fertilised oocyte. That is, the cells have become smaller. In humans, the blastocyst is around 100 cells, which is 6 to 7 rounds of cell division. At this stage in humans, the blastocyst is also still the same size as the original oocyte. It's worth noting that the blastula and blastocyst are different. A blastula is a hollow ball of cells, whereas a blastocyst, which is still essentially a hollow ball of cells, has inside it some differentiated cells. Plus, blastula tends to be used for animals, whereas blastocyst is used for mammals. The next stage of development is gastrulation, the formation of the gastrula. During gastrulation, we get the formation of structures and the organisation of cells. The ball of cells invaginates and we get the formation of the mesoderm, the middle layer of cells or tissues of an embryo that goes on to form cartilage and muscle and bone, the ectoderm, the outermost layer of cells which include the epidermis, nerve tissue and nephridia, the endoderm, the innermost layer of cells which goes on to form the lining of the gut and associated structures. And it is the VegT messenger RNA I mentioned earlier that produces a protein called VegT that activates genes that code for the cell to form the mesoderm and the endoderm. And this is an example of the role of transport and localization of messenger RNA in a cell, because if the VegT isn't in the right location in the oocyte, then we don't get the correct development. So what about the fate of the cells? How does a cell become a muscle cell? Why does that cell always become a muscle cell? Is it because it inherits a particular cytoplasm during division from the parent cell, or is it down to position in the developing embryo? Well, in the developing Xenopus blastula, we can inject a cell, a blastomere, with a dye and see where that cell and its daughter cells end up in the developing embryo. We can do this with cells in a particular position or to cells we have moved to a new location in the blastula. And when we do this, we find that the final fate of the cell is due to the location in the blastula. In fact, we can use this approach to map the cells in the blastula to their final role in the frog. And when we do this, we find the following arrangement: epidermis and nervous systems at the animal pole and everything else at the vegetal. There is also a dorsal topside and ventral underside arrangement in the blastula. The way this all works is that the cells come under the influence of the protein secretions of their neighbouring cells. And these secretions drive the production of tissue-specific transcription factors. However, you should be aware that as the organism develops, the cells lose their ability to become different cell types. They start off as being totipotent, that is, they can become all cell types. The cells then become pluripotent, that is, they can become most cell types. Next they become multipotent, that is, they can become several cell types, before finally becoming specialised cells, that is, terminally differentiated. But this specialisation can be reversed. Just think of Dolly the sheep.